Good morning, everybody. It's a great pleasure to be here. This is the second time I give a talk, I think, this institute, and it's a great pleasure. I would like to talk to you about stochastic dynamics. In many ways, this is a very natural progression from the previous talk on deterministic dynamics, but I will also try to show you the connection between the two, and there's a very important connection. And in doing so, we will look at various examples, a lot of examples from synthetic biology, some examples from systems biology. And I will also try to cover some of the background mathematics that is needed for modeling stochastic processes. So this is the outline. I will talk about cellular noise and how to model it. We'll look at feedback regulation in the presence of noise, both at the population level and at the single cell level. So this is the multiscale aspect of my talk. And then we will look at oscillations and entrainment in the presence of noise. And one of the things that you will find, and it's, I hope, a running theme throughout this presentation, is that whereas, for example, in many engineering applications and physics applications, noise is something like a nuisance to be avoided, in biology, we are seeing more and more examples where noise is actually something that introduces new functionality that is beneficial, okay? And so I'll try to give you at least two examples of that. And it's kind of striking and it's interesting. And it's the way the noise interacts with the dynamics that gives rise to these phenomena. Okay, so let's start by looking at gene expression. So we have seen this in the previous talk. And this is a diagram that I will keep referring to. We have transcription into mRNA, and then degradation, and then translation into a protein. In this case, we will look at a fluorescent protein. Let's say green fluorescent protein, which degrades at a particular rate. But if you look at a culture of bacteria that has a system like this, and then you try to look at the fluorescence, you see that some cells are fluorescing more than others, okay? Whereas the old dogma is that you evaluate this as a uniform mixture where you look at populations. At the single cell level, there are very important differences, right? And these things can be quantified. So for example, if you run these things through a flow cytometer, which essentially shines light at each one cell at a time as these cells pass through a microfluidic channel, and then measures the intensity, and then you can draw a histogram, which gives you an idea about the distribution of these proteins. You can see something like this, right? So even though these are genetically identical cells, the expression of various cellular proteins is not identical. And this is very significant, and these differences can actually give rise to very important biological phenomena. So it's important actually to try to understand where they come from and how one can model them and deal with them mathematically, okay? So if you look inside a cell, it's a very busy place, a very packed place full of molecules that are moving around due to their thermal energy. And reactions take place in the cells when two reacting molecules collide with each other, right? And there are three different factors that conspire to give rise to this cell-to-cell variability, or this randomness that I was talking about. One is that the molecules aren't actually continuous variables, so they come in discrete numbers, right? So 1, 2, 3 copies per cell. And very often, this number is quite low. So you have maybe one copy of a gene, or maybe a handful of mRNA copies. And so and the last part is that reactions inside the cell are by their very nature random events, right? The timing of these reactions and the order at which they occur, precisely because of the randomness inside the cell. So all of these things will also be random, right? So those three things give rise to stochastic chemical kinetics. So if you look at individual cells, for example, a particular protein copy number, over time it may look like this. A neighboring cell with identical genetic makeup may have something different, and a third one may look more different still. So a deterministic model would represent these things as one trajectory, right? And so if we want to capture this variability between cells to cell, one cell to the next, and even variability within the same cell over time, we have to look at a new paradigm that allows us to do this. And that's a probabilistic paradigm. That's the paradigm of stochastic chemical kinetics. Okay? So how would one go about doing this? Let's look at our favorite system, try to see how you would model this stochastically. We look at the abundance of proteins and mRNAs as discrete random variables. So this is how we represent them. In this case, X sub R for the RNA and X sub P for the protein. And then reactions are random events. So the probability that a single mRNA, for example, is transcribed here in a given time h is given by the transcription rate times the time increment plus the term of order h, right? And similarly for degradation here. And so this gives you a transition intensity, if you will, for this reaction. So in this particular case, XR is just a birth-death process, stochastic process, that can be represented like this. I'll come back to this expression later on. But the important thing, because it's a stochastic process, we can talk about its mean and variance and various statistical properties, right? And in this particular case, let's say if you look at the evolution of the average for the RNA, it's given by this formula, Kr minus gamma E of X of R, expectation of X of R, and similarly for the protein. Now, interestingly, you will notice that these dynamics are exactly the same dynamics you would have obtained had you written these equations deterministically, right? So if you followed Mark's lecture, previous lecture, these are the dynamics that would arise, right? So in this particular case, the dynamics of the mean happen to coincide with the dynamics, deterministic dynamics that you would get from standard mass action kinetics. Now, it's very important to emphasize that this is not always the case, right? So this works because these are monomolecular reactions that don't involve two things coming together. But as soon as you have two molecules coming together to form a reaction, then this breaks down. So even the mean, dynamics for the mean don't necessarily coincide, and usually they don't, with the dynamics you would obtain if you were to write the deterministic equations from reaction rate equations from mass action kinetics. And so fortunately, in this case, they're the same. But the interesting thing is that with this framework, now you can view each cell as a different sample path of that stochastic process. And then it gives you an explanation why identical cells could have, at a given time, different copies of mRNAs or different copies of proteins, right? So these are different trajectories or different sample paths of the stochastic process. For this simple gene expression circuit, because we can write the dynamics of their mean and variance, we can look at the stationary solution for these equations, and we can get explicit analytic formulas that tell us how measure the variability, right? So for example, the mean for the protein is transcription rate times the translation rate divided by the product of the degradation rates. And the coefficient of variation, which is the standard deviation divided by the mean, is a measure of variability, is given by this expression. For the RNA, this expression is simple, but you would notice that this coefficient of variation goes to 0 the larger the expectation of XR. So the more mRNAs we have in the cell, the less the fluctuations matter, okay? Which is an important thing. So if you have a very large copy numbers of various molecular species, maybe you don't want to look at a stochastic model, and a deterministic model should be adequate. And I will give you an example of that. Similarly here, if you have, so this here is actually turns out to be a Poisson process, so the CV is 1 over the square root of the mean. In this case, this is not a Poisson process, but similarly, if the expectation, if the average number of proteins is high, the coefficient of variations becomes small, and then maybe you want to look at a deterministic model. So this is how you would do this for a very simple gene expression model. But it turns out this approach, where you would model these things as stochastic processes, discrete random variables, and so on, generalizes to any set of chemical reactions. And I think it's important for me to show you, just to give you a flavor. Maybe you've already seen this in previous lectures, but you can do this in general. So if you have a system, let's say a cellular system with n different species that you're interested in, each one of them would be a discrete random variable. And then now you have a discrete random vector, x1 up to x sub n. Let's say you have m reactions among these n different species. The reactions are R1 up to Rm. And every time a reaction fires at a given moment in time, the number of copies of the various species changes according to the stoichiometry of that reaction, right? So for every reaction, there corresponds a vector that indicates exactly how the abundance of these species will be changed after a single firing of that reaction, okay? So maybe one molecule is incremented by 1, another one is decremented by 1, and so on, right? And since we have m reactions, this is a matrix. You can stack this in a matrix, a stoichiometry matrix, which has m columns. And the last ingredient you need is a transition probability. So how frequently these reactions happen. And so if you're in a state x, a little x here, the probability that the kth reaction fires in a given time increment h is given by some function of the state that reflects the reaction rate, right, times the time increment plus something of order h, okay? So this here is usually called the propensity function. And just to give you an example, this is really very simple to do. Suppose we have these three reactions. We have a birth event of a species we call S1. There's another species S2. When S1 reacts with S2, one copy of S2 disappears or degrades, right? So maybe this is an enzyme of some sort, right? Or a protease. And so a third reaction would be perhaps a natural degradation of S1. So for this reaction, the stoichiometry vector would look like this, because you have one more molecule of S1 once the reaction fires. S2 doesn't change, so we have a 0 here. And the rate of this reaction is constant. For this here, this is a bimolecular reaction. And you probably can't see this green on this background. But OK, I'll show you. I'll just tell you what it is. So the stoichiometry matrix is vector 0, negative 1. S1 doesn't change with this reaction, but S2 is decreased by 1. And the propensity function is actually a product of the population of x1 and x2, okay? Of course, that's natural. You know, the more of S1 you have, the more likely that reaction will fire. Similarly for S2, and actually the rate depends on the product, okay? And this is where you get these bimolecular reactions that give rise to the nonlinearities and the difficulties in dealing with this stochastic system. And the last reaction, of course, is straightforward. So with the degradation, you have -1, 0, and this is just a linear term. And so if you look at this, this is nothing but a continuous time discrete state Markov chain. In this case, we have x1 and x2. We have two species. And so if R2 fires here, then followed by R1, R1, R2, and so on, then the trajectories follow what you see here. So piecewise constant trajectory, right? Okay, so this is, in a nutshell, stochastic chemical kinetics. There are different ways to look at this process that I described to you. Each one of them is useful depending on, you know, what kind of question you're trying to ask and how much time you have you're willing to solve these things, okay? And they also present some interesting mathematical challenges. And this is very much an area of research, much less developed than the deterministic chemical kinetics that you know. So one way to represent these things is using what's so-called random time change model, right? So this is a clean way to write these stochastic processes. Think of these as vectors. So this process at any given moment in time is given by the initial value of x plus a sum of Poisson processes, unit Poisson processes. These are the stoichiometry vectors. And what's curious about this is that the argument of this unit Poisson process is itself a function of the Poisson process, right? But because these are piecewise constant things, actually it's very easy to simulate something like that on the computer. And that gives you a quick and fast way to generate sample paths, okay? This is very much like the Gillespie, Dan Gillespie, or the SSA, stochastic simulation algorithm of Gillespie, right? By simulating this here, you get as many sample paths as you like, and then you can compute statistics for those. So this is a direct representation. It also has very interesting, useful mathematical properties that one can use for proving various things. Another one which is also very useful is looking at the dynamics of the moments. For example, the mean or the second moment here and so on. And it turns out, it's not surprising, that the dynamics of the mean and the variance and so on, the third order mean and so on, they follow or they evolve according to deterministic ordinary differential equations, right? Just like we've seen in the case of gene expression, these are deterministic ODEs. Here also, you can write these things down, and they are deterministic ODEs. There is a catch, though, and it's a big catch, and that's the following. So suppose we want to figure out the equations for the mean for even the reaction we've seen, the example we've seen in the last slide. So this depends on W. This is the propensity vector. And so the propensity vector for one of the reactions is the product of x1 and x2. So we have a quadratic term, right? So here we have an expectation of a quadratic term, so that's a second order moment. That means to get this term here, you need to go to this equation, which has the second order moments. But if you look at the right-hand side of this equation, so this here, this is a vector form, but you can imagine what it would look like. This would be a second order term multiplied by x, so you have a third order moment, right? So in order to solve for the second order moment, you need to know what the third order moment is, and so on and so forth. And so you end up actually with an infinite set of linear or of ordinary differential equations that you cannot solve. Okay? So this is called the moment closure problem. So the moments are not closed. Happily, when the reactions are monomolecular, so when you have, for example, affine linear propensity vector, that does not happen. Like in the case of gene expression, this is not an issue, right? Because we don't have any bimolecular reactions, right? And so keep that issue in mind. It does form a significant challenge. Very often what people do is they say, okay, well, we think that the third order moments, or the distribution looks more like a log normal. And so with the log normal, you can write an expression of the third order moment in terms of the first and second order moments, and you can plug that back in, and then you have a closed system, right? But that's an assumption that, depending on its validity, could give you some errors in the dynamics. And there are various moment closure methods out there. There's at least 4 or 5. And they often are valid in different regions of the dynamics. The other way to look at this stochastic process is try to say, okay, well, how does the probability distribution evolve as a function of time? Okay, and the probability of being in a particular state at a given time, so this is the PDF, also follows a differential equation. Actually, it's a linear ordinary differential equation as well, right? And this is like the forward Kolmogorov equation for this process. And it's often called the chemical master equation. So here also, there's a lot of challenges. Because, in fact, if you write this vector P, it's an infinite vector. And even though these are linear dynamics, you're evolving over an infinite state space. So you have an infinite integer lattice that you're dealing with. We had developed, and others developed, methods for truncating the state space and trying to solve a finite linear system while providing an error bound on, the truncation. So that if you can truncate, for example, in a particular region of the state space, you can figure out how much probability you're missing, how much error is being introduced by this truncation. But this is still a very challenging problem, because even when you do this truncation, you still end up with a huge linear ODE to solve. So for small problems, this will be a good approach. For larger problems, this could be hopeless. And finally, there's various stochastic differential equation approaches to approximate these dynamics, right? So one of them is I guess this is called the Van Kampen. This particular is called the Van Kampen approximation. There's also diffusion approximations and so on. And those you can solve numerically. They're faster than this, but they're still approximations, so they don't necessarily capture, for example, bimodal behavior very well. But if you have fluctuations around some mean, then maybe this is OK. All right, this is a whole area on its own, right? So I just want to give you a flavor for what you can do, and depending on the quantity that you're interested in, you have different dynamics that you can solve. But this is SDE, this is deterministic. This is just numerical Monte Carlo simulations. Now I would like to come to this very interesting and important result, and it actually is not very difficult to prove. And that is the following. So I've described to you the stochastic chemical kinetics, and the previous speaker told you about deterministic chemical kinetics, the ODEs. Well, how do these two worlds relate to each other? I think that's the important question to answer. Certainly, one expects consistency between these two systems, right? And so this result gives you exactly this, right? So let's now take our stochastic process x of t, and this is copy number, since in order to try to relate it to concentrations, which is how the reaction rate equations are usually written, we divide by the cell volume, right? So this is copy number per unit volume. And now this is a measure of concentration. And let's say you write down the deterministic reaction rate equations, and that's x of t. And so now it's fair to ask, how does this stochastic process x of omega relate to the deterministic solution x of t? All right? And so here's the answer to this. So if this is a deterministic solution for the reaction rate equation, let's say you start the initial conditions at x naught, and this is the stochastic representation of the same reaction system, and you also initiate it at the same initial condition, then one can show that, let's say over any finite interval, all right, from 0 to t, as you take omega to infinity, as the reaction volume increases to infinity. And when I say the reaction volume increases to infinity, you must always still respect the fact that the initial, as you increase the volume, that the initial condition is the same, right? So as omega goes to infinity, this stochastic process converges. Actually, it converges in the strongest possible way. It converges almost surely to the deterministic solution, okay? And this is just the a consequence of the law of large numbers, actually. So it's not difficult to prove. And so all of these fluctuations, as you increase, they start getting smaller and smaller, and eventually the trajectories will all collapse to the deterministic solution. But until that happens, very interesting and funny behavior can take place, okay? So this is only in the large volume limit that the two coincides. If you don't have this large volume limit apply, you still have all of these interesting dynamics that I will describe them today, which may have no counterpart in the deterministic world. Okay? Okay, but keep this in mind. This is sort of a sanity check. This is what allows us to say the two representations are actually related. Now, in practice, it's difficult to say, well, how large is omega? Okay, so this is just a limit result. It's not an exact solution. Okay, so now let's talk about two problems that we wanted to discuss. The first one is regulation, and again, I couldn't have had a better introduction than the previous talk with feedback loops. So before I was doing biology, I was a control engineer or a control theorist, and so feedback is at the heart of control, right? So you'll be hearing a lot about, in particular, negative feedback. Not so much positive feedback, but negative feedback, because we do like our systems stable and have a fixed point, follow a particular fixed point. So here's an example that comes from at least my past. So we have an airplane dynamically, you'd like to control it to a particular altitude. And so a typical feedback control mechanism looks like this. So you first have to measure the quantity you want to regulate. In this case, the altitude. So we have some sort of altimeter. And then the pilot has a desired value for the altitude. Let's call it R. And then the two variables are compared, and the difference is the error, which you would like to be 0, right? Now, this error is input into the control system, which executes a control algorithm. the output of which is a command signal that is issued to the elevator surfaces, flight surfaces, right? So if you want to go up, these things, you increase the angle of attack, and then the altitude goes up, increases, and then vice versa, right? And so you want this feedback system to be stable so that it results in a fixed point, just like we've seen in previous talk, and the fixed point trajectories will converge to that fixed point. A very typical control algorithm, although this is not what's used in most airplanes, but some aspects of it are, looks something like this. The input U here is some function of the error. And what is that function? It could be a constant times the error. But that would be very naive. That would be proportional feedback, and it's not enough to do good control. The other interesting piece is this one. So you also want to add to this term an integral feedback term, right? So the U is actually some function of the integral of the error. It gives you an idea of the past history of the error. So if you had an error for a long time, then this term would be large, and it will speed up the control. If it's just if you just recently started to develop this error, this term would be small. It wouldn't have much effect on the dynamics, right? So this here tells you more about the history. This tells you more about the current value of the error. And this is a prediction term that gives you an idea about where the error is heading, the future, right? So a combination of these three terms is usually used. This is called the PID control system. I'm sure many of you have heard of them. How many of you have seen or heard of PID controllers? Okay, almost everybody. Okay, so that's what they do. And they're very nice, very simple. You just have to tune 3 parameters, and then you get your dynamics. Now, interestingly, the integral term is particularly useful because it has the property that it gives you 0 steady state tracking errors, right? So it ensures that the error goes to 0 in the steady state, okay? Why is that? Well, if the error doesn't go to 0 and it goes to any other value than 0, then this integral term will build up, and U will go to infinity, which will be an unstable system. So if you can stabilize the feedback system, you're guaranteed that this error goes to 0 asymptotically, right? And so it's very interesting to see, actually, the examples I will show you, synthetic feedback circuits that we want to build in this integral feedback term. And of course, if you look at E. coli or look at the way your body regulates calcium, for example, integral feedback is very much present in the dynamics. Okay, so this is just one example. So now I would like to apply this feedback control or feedback regulation to living cells to try to control gene expression. And what I'll show you now is actually something we have built, and I will describe to you the details. In order to do the feedback control, you have to be able to actuate. You have to give commands to the system, right? So since we are coming from the outside world, wanting to control these cells or gene expressions, we have to have a way to talk to these cells, right? In this particular case, this is going to be light, okay? So we're going to use optogenetics to do this. So here we have a system that comes from Jeff Tabor's lab. The idea is that if you shine green light, this CCAS is a histidine kinase, autophosphorylates and then passes this phosphor group to this response regulator, CCAR. Once phosphorylated, it dimerizes, and that acts as a transcription factor that Mark had mentioned before. So it binds to the promoter site and turns on gene expression. And now the gene in this case is just a fluorescent tag, right, GFP, so which we can measure, right? And when you shine red light, the opposite process happens. This acts as a phosphatase, or at least it's thought to act like a phosphatase. The exact science is not completely understood, but in any case, the transcription factor comes off, gene expression is turned on. So now we have a way to actuate these cells and hopefully try to control gene expression. This is the setup. Okay, so we have a system that looks like this that regulates the turbidity of the culture, and then we take automatic samples every so often that we pass it to the flow cytometer, which gives us an indication of the distribution of these of the fluorescence, as I told you before. The output of the flow cytometer is then fed back to a computer, which has a set point that the user determines. And then a control algorithm is running inside the computer to determine the output of two LEDs, red or green, thereby closing the feedback loop. Okay? So this is an optogenetic feedback system designed to regulate gene expression. And so yeah, this is the output. You have these distributions. I mean, you don't necessarily need to use all the distributions. You could just try to control the mean, in which case you just get one value, right? It looks something like this when the green light is on, and this tube here has all the cell culture in it. So notice this is population control, right? So I'm controlling the entire population. I'm not targeting individual cells. I'm just looking at the whole population. So it makes sense to look at the expectation or the mean as a measure for gene expression in this case. And that I don't even need a flow cytometer for this. I can just have a fluorimeter that measures the bulk fluorescence, which gives me an idea about the average gene expression. Okay, very good. So the dynamics, since everything is the expectation, so we're dealing with deterministic systems. So this is the expected value of RNA. That's the protein. Okay, so it takes some time for the protein to mature and to become fluorescent. So I have another state, a third state. This is the protein, the not mature, immature GFP, and this is the fluorescing GFP. Okay, and if you look at these dynamics, they're exactly linear dynamics, right? It's just gene expression. And so it's not particularly hard to control them with this input U here. Right? This is just a change of variables so that you can have in terms of fold change rather than absolute concentrations. But in any case, to be able to control this system, I would need to know something about the parameters. So one can run input-output experiments and try to do system inference or model parameter inference, and come up with these dp, km, and so on, which we've identified over here. Now, the important thing about modeling when you're trying to do feedback control is that the model need not be accurate. And actually, that's an important point. So when you do modeling for control, it's not like you're doing modeling for systems biology or trying to understand the process. You're just trying to get an idea about the dynamics, and then the feedback takes care of any mismatches or discrepancies between the actual model and the one that you have for modeling. So if you capture, for example, the steady state value or the DC value, maybe you can even approximate it by first or second order system. Very often this is all you need to do, even if the dynamics are much more complicated than this. So here's the system. That's our light system. You've got fluorescence, just like the flight control. We have an error, which is the difference between the desired fluorescence and the actual one. And we have a control system. In this case, we're going to use actually two different types of controllers, a simple one and a more advanced one. The simple one it's not even a PID. It's just a PI, proportional integral. Now, significantly, it has this integral term, so you would expect it to have these nice steady-state properties. And so here are some experimental results. So the red is the proportional integral controller. The green is a different kind of controller, which I haven't described yet. It's called the MPC, or Model Predictive Controller. But let's come back to this. For now, you should notice that for all of these three different set points, you can see that the fluorescence is following very nicely, right, with zero steady state tracking error. And if you look at this here, the red, for example, that's the light, the control signal. That's the LED intensity, right? So we actually keep one light constant, and we vary the red. So we keep the green constant, and we vary the red. Yeah. Time, yeah, sorry, this didn't show up. Yeah, this is time in minutes. This is not an airplane dynamic. It's very slow. So this could take several hours. Yeah. So that's time in minutes. I tell my students they should label all the axes, and I was so fed up with this that they don't do this. I started charging 5 francs every time somebody doesn't label an axis. So now I'm on TV doing exactly the same thing. Okay. Well, I owe somebody some francs. Okay, so this is time in minutes. And, oh, no, also here, it's the same. So the interesting thing is that, so the model predictive controller is actually a different kind of controller that solves an optimization problem every time instance. So it has a model of the process, and it looks ahead a certain number of steps, and it says, okay, if these are the dynamics, and if I want my output to look like this, it solves an optimization problem to see, what the input should be in order to get the output to match what the model would give. And then it would execute one time step, right? And then repeats the same thing. So it increases the horizon by 1, and it repeats. So this is quite successful in the industry, and actually it has a lot of nice properties, but it's much more complicated to implement than a simple PI. Now, in this particular case, the set point is oscillation. It's a sinusoid, and you can see that for the model predictive controller, it has no problems matching it, but the proportional integral controller breaks down. It just cannot follow, right? So you need the more advanced controller to follow sinusoidal signal. Now, interestingly, you take this control input that so successfully tracked the sinusoid, then you apply the same exact input the next day on a culture prepared exactly in the same way without the feedback. You just apply the same light sequence that worked the previous day in open loop, and you see that very quickly you start developing an error that increases as time goes on, which basically suggests that you actually need the feedback action. You cannot just apply the input open loop, even though the same input worked the previous day. Because, from day to day, there are all kinds of variability. Temperature might be slightly different. Maybe there's a slight difference in the way this was prepared. Maybe things stayed in the tube a little bit longer, so they're not identical systems, right? And so the feedback takes care of all of this. So just to show you how well this works, and one of the reasons why you want to use feedback control and why nature uses feedback control in many of the systems is that it's very robust. Right? So here we change the temperature from 37 to 28 degrees, which is a big change. And without the feedback control, you will have a steady state error. This is for the MPC, I believe. Without it, you don't even notice this. And notice that now the control signal actually just simply adjusts to the new temperature. Okay, so I think that's all I wanted to say about this particular system. We can apply feedback loops externally to biological systems. I mean, that's one way to do it. That's what I would call in silico control. But that's not very useful if you want to do individual control, right? So one way to do individual cell control is to do this under the microscope, and we've done this to try to shine light on each individual cells and try to do something similar. But another way, which is actually much harder, would be to build in or genetically engineer a feedback loop into living cells. Okay, this is just a cartoon. So each cell has its own little feedback controller that you would introduce to do a particular task. Okay, so I'm going to skip this part for the interest of time. This tries to compare the variance, the noise for different methods. And I would like to now just focus on this single cell feedback regulation. And this will also tie in with the previous presentation. And so I would like to describe to you a new controller in E. coli that actually we're assuming that we have large copy numbers of everything, so we can even do the deterministic representation, just like the previous talk. So in this case, here's our system that we want to control. It's simple gene expression, transcription, translation. And this here is LAC-I. Okay? The protein is LAC-I, and it's labeled with a red fluorescent tag. Now, this thing here tetramerizes, so LAC-I tetramerizes, which is nice because it gives us cooperativity. And then this is supposed to feed back on another gene, and this here is a tag promoter that is repressed by LAC-I, But this gene actually makes error C, which is tagged with a green fluorescent protein now. Okay? And the error C activates this gene circuit. So this is a simple feedback loop. It's a negative feedback loop because the more LAC-I you have, the less production of error C one has. And in order to be able to have a knob, in order to dial in the set point, we have this IPTG, which was also mentioned earlier. It's a sugar that is not metabolized by the cell, so it's a good way to adjust this repression, right? So now we have a simple feedback system. And then just to test its robustness, we built in yet another module that adds a disturbance. So this produces more LAC-I. And if the loop is working properly, I should be able to reject this extra disturbance, and I'll have my LAC-I, which is the output, track the fixed point that I want. Right? So this is the idea. So think of this as a tunable disturbance, okay, that you can add in. Okay. And then this is open loop without this tunable, without the feedback, just for comparison. So again, this is all deterministic right now because we're assuming LAC-I molecules is quite large. So we're looking at a concentration of LAC-I molecules, similarly for error C. And so we look at the deterministic representation. And again, you know, so in this particular case, so we have a synthesis rate. We don't have mRNA modeled, so we actually do time scale separation. We just look at proteins here. It's just easier to represent because if you want to describe these things on a nice phase portrait, as you have seen in the previous talk, 2D is actually the way to go. Of course, you can always simulate in 4D. So in this case, so you have this transcription or synthesis rate. We have this degradation. In the case of RC, we actually have a tag, a degradation tag. So we have active degradation as well as the exponential degradation here. And because of this tetramerization, so we have a Hill coefficient roughly of around 4, right, for this system. So again, to look at the dynamics, this is the phase portrait. So this is Y, the output. This is U. That's RC, and this is LAC-I. And so you look at these two knock lines, and the point in which they intersect is exactly the fixed point. And in this case, the fixed point is stable, so all trajectories will converge to this fixed point. Okay. As you adjust the IPTG, what actually happens is that you shift this thing around to the right. And so this allows you to get different intersection points and therefore different values for the output Y, right? So this is like the tuning knob. And interestingly, what happens is that as you add a disturbance, that's equivalent to shifting this curve to the right. And so the idea is that if this thing here is sufficiently steep, then the intersection point, at least its axis, y-axis, wouldn't change very much. And so you would achieve good disturbance rejection, right? So ideally, this would be vertical, which is what you would get if you have an integral feedback controller. But in this case, if you just have high gain, you will be able to get small delta Y, okay? So that's the basic idea behind the dynamics here. Oops, okay, hit the wrong button. So, okay, so we built this in my lab, and then I want to show you these step responses for E. coli, right? So these are different values of IPTG. Again, this is here by, okay, it's minutes, so it's labeled. So this is in minutes, and you can see the step responses for different amount of IPTG. You can clearly see these things are actually very nicely tunable. And corresponding to different value of the output, you have a very different value of the control signal, right, as you would expect. So this is a feedback loop that is certainly working. And in this case, also, you can try to see what would happen if you start different cultures at different, so you have different initial conditions, and you put them in the same culture with a new set point, you find that all of them will actually converge to the new value, right? So it doesn't matter what the initial condition is, you will always converge to the value that corresponds to the IPTG level that you have. Okay, and I don't want to spend too much time on this. You also have very good disturbance rejection properties for this system. because of the way it was designed. And in fact, the higher the Hill coefficient, the better the disturbance rejection will be, right? Because the amount of error depends on the steepness, if you will, of the Hill curve. Okay. This took a lot of work and at least 2 or 3 years to do. And we had all sorts of problems. This is just a subset of these problems. So when I say it took 225 plasmids, what I really mean to say is that there were 224 failed experiments. And each time you want to change a parameter, it takes a week, to rechange that parameter. So unlike the in silico control where you can just go to the computer or MATLAB and then change the number and hit return, this is much harder because you're actually changing these genetic parameters. A big problem that we have faced is the loading effects. So if you make too many proteins, then this has a metabolic burden on the system, and it affects the growth rate, and the cells can become sick. So you cannot make too much protein, but at the same time, you want to make enough so that at least you stay in a regime that you can actually observe these things. So motivated by this, we had ideas, okay, well, if you want to do feedback control is really about information processing. It's not about energy or material. And you ought to be able to do information processing with very, very low copy numbers, right? And so this brings us to the next part of the talk, which is how do you do feedback in the stochastic regime when you have very low copy numbers? And that's where we want to move. Are there any questions about the previous part before I go onto this part? Okay, so again, we've discussed this. You have a set of chemical reactions. So now we want to work in the stochastic regime. We have to deal with stochastic chemical kinetics. And suppose we build a feedback control system, or maybe look at an endogenous feedback loop. One way to ask the question, so in the case of the deterministic world, we say, okay, we have a fixed point, and we want the fixed point to be a stable fixed point. So the trajectories actually converge to it. So what is the equivalent notion for the stochastic world, right? So what we found is that the most useful one by far is the notion of ergodicity, okay? Which basically means that regardless of the initial distribution of the various proteins, there's always a unique stationary distribution that it converges to, right? So there's a unique stationary distribution. So we will call a reaction network ergodic if there's a unique PDF pi such that any initial distribution converges to. So the solution of the chemical master equation, if you will, is unique, okay? Okay, and here is a sort of mathematical formulation of the feedback control problem. Given a set of stochastic reaction networks, this is this uncontrolled system, augmented with a new stochastic reaction network, which is our controller, such that together the resulting network of reactions, the closed loop, satisfies this ergodicity property. But it's not only ergodicity, we also want some more. We want a particular output of interest, let's say the nth one, just to pick one, to converge to some desired set value, set point, right? So let's say the nth variable here, and it could be anything, you can label those, on average converges to the desired set point. So that's stability and tracking, okay? Now, the way it's formulated, it basically has a synthetic biology twist to it, but it doesn't really have to, right? You can look at an endogenous circuit, and you can look at and this would be the closed loop, and you can ask, is it ergodic, and do the outputs converge, and are they guaranteed to converge? And obviously, we want this to be a robust property, so that if you add disturbances to the system, or if you change some parameters, you're still going to converge to the same R, right? So this property should be inherent, should be robust. It shouldn't depend on the dynamics or on the parameters okay. So we've come up with actually a motif that seems to do the job very nicely, and I just want to describe it to you now. It's a very simple motif. that is bimolecular. So suppose you have a network of chemical reactions that you want to control. Let's say you would like this xn, just pick on one variable, this variable here, to approach R on average, right? And of course, you need to have something to control it with, something that you can wiggle. So let's say this is x1. So by adjusting x1, we want xn to follow a particular trajectory, right, on average. And so in order to do that, we will introduce a control system, right? And here's a control system. It's a molecular controller. And this controller has basically 4 reactions and 2 species. And I want to show you what those are. So just for lack of any other name, we'll call these species Z1 and Z2. Okay, they could be anything. The rate of production of Z2 is actually dependent on x sub n, on the variable that you want to measure. So this here acts like a sensor, right? It measures the amount of xn, and the abundance of Z2 in some way actually depends on how much xn is. So this is a sensor of xn. And now Z1 is actually produced constitutively at rate mu. And there's this annihilation reaction that takes Z1 and Z2, and they bind together irreversibly, okay? Now, this is exactly the same system that was shown in the previous talk, this ultrasensitive A and B, I think you called them, Mark, right? So A and B. So these things bind together. They could be irreversible, but at least the binding affinity is much stronger than the off rate, right? So the on rate. Now, Z1 acts as the actuator. So depending on how much Z1 there is, so the more Z1 there is, the more X1. So that closes the feedback loop. Okay? So think about how this would work, just intuitively, right? So if you have a lot of Xn, then you have a lot of Z2. And if you have a lot of Z2, that will sponge out the Z1 from the cell, and you have less Z1. If you have less Z1, you'll have less X1, and so therefore you will have less Xn. Okay? And similarly, the other way around, right? So if you don't have enough of X1, you have a small amount of Z2 that leaves Z1 to activate X1 and increase the level. So at least intuitively, this makes sense. But it turns out that this system actually has much, much nicer properties than just that. So deterministically, it works nicely, but it has a very intriguing stochastic properties. And these results are actually captured in this one theorem. I'm probably not going to show any other theorem, but it is an honest-to-goodness theorem, and it does tell you a lot about this system. Okay, so basically this is saying that suppose this, it's basically validates that this controller works well in the stochastic setting. It says suppose this network is it's irreducible, and you look at the dynamics of the mean for that network, and suppose those dynamics are stable. So this matrix is, the Jacobian has eigenvalues in the left half of the complex plane, right? And if you apply to this system this specific controller, regardless of what's inside, this network the closed loop is guaranteed to be ergodic, so we have our stability result. But also, the first and second order moments are uniformly bounded and converging, okay? And finally, we have our tracking property. In other words, the average value of xn converges to mu over theta, which is the set point for this system, right? So mu is this value, and theta is that value. So you have to be able to somehow set the ratio of those two. But notice it doesn't depend on eta, it doesn't depend on anything in the network. So if you just set mu over theta, then you're guaranteed that xn is going to converge to the proper set point, right? Okay, so I mean, if I were to prove this deterministically, I would have to use something like Lyapunov's theory to come up with a Lyapunov function that is positive definite, whose derivative along the gradients is negative definite, and therefore proving stability. They're similar tools, but they're quite a bit more complicated. In the stochastic case, you have to use Foster-Lyapunov equations to do this. And you apply the generator to this function v, and you show it exhibits certain properties. And so the nice thing is that you only have to do this once. It's important to say that this theorem, as it stands, is for networks that are basically affine. If the network it doesn't have affine propensities, and you can guarantee ergodicity, then it doesn't matter whether it's affine or not, then you still have the same results. So, but this condition allows me to get ergodicity. Okay, so let's apply it to our gene expression circuit and see how it does. In this particular case, it's the same as before, and we just have standard gene expression. This is our integral controller, and we call this antithetic integral controller because these things move in different directions. And there's another. Okay, so this result says that regardless of the gene expression parameters, Kr, gamma r, Kp, gamma p, and this eta, this network will always be ergodic, and also the expected value of the protein will always converge to mu over theta, right? So very, very robust property. Okay, how much time do I have left? I just want to make sure I get to the 30. Okay, all right. Here are some simulations just to show you that this is the case. So we've picked some parameters for this system, and the dashed line is the set point. That's mu over theta, right? And so this is about a few hundred Monte Carlo simulations. And so indeed, the average for the expected value of x2 goes up. It was a bit overshoot, and then comes back down and reaches a set point. If you change the set point, it does something similar. Change the set point, it does something similar. But the steady state properties are always guaranteed. And the more Monte Carlo simulations you have, of course, the smoother this is. Now, I should say that we don't have a closed loop solution for this because of the moment closure problem that I mentioned earlier. After all, this reaction here is bimolecular reaction, and so that screws up the moment closure problem. So we have to rely on these simulations. What's going on with the other variables? Well, z1 is here, z2 is there, and x1 is somewhere in between. Interestingly, if you look at the single cell behavior and see what's going on to the protein level, it's very, very noisy. So this here is x2, and it's all over the place. If you look at the control signal, z2, it's even worse. It's going between 0 and 1 molecule per cell. Okay, so very, very low copy numbers. So the control system itself is not high copy number. It's just 0 or 1 molecule. Sometimes it's 2, sometimes it's 4, sometimes it goes to 6. But you know, it remains in this very low copy regime. And in fact, it's just the duty cycle that affects the control effort, right? So if this is mostly 0, then the control signal is low. If it's often 1 or 2, the control signal is higher, right? So this system is adjusting the control not by increasing concentrations, but by increasing the duty cycle, or the amount of time it spends in the 1 or 2 copy number. Okay, so this is extremely noisy. When I first saw this, it reminded me of something else we had looked at almost 10 years earlier, and that is the heat shock response in bacteria, where the main sigma factor, which controls the heat shock response, called sigma 32, is also present in very, very low copy numbers. In fact, this figure comes from that work. It goes from 0 to 1 molecules per cell. Okay? So it would appear that at least bacteria employ a similar kind of control system. Okay, how about robustness? So here's the robustness test. So what we do here is we take Kp and we just abruptly changes the parameter, let's say, to twice the value. So Kp goes from 2 to 4 over here. And what you see is that, of course, when you change the parameter so abruptly, the dynamics will change. You have new dynamics now. And so you'll have transients. But then the variable of interest, which is this one, goes back to the right value, as it should, as guaranteed by the theorem. But the other variables take on different values. And they take on whatever different values are needed in order to ensure that x2 is at the proper value for these new parameters. Okay? That's already built in. That's, in some sense, the function of this integrator. Similarly, you change gamma p, same story. you change gamma r, again, so transients. But look at Z2 here. It actually increases more than double, but X2 is exactly the same where it needs to be, okay? Okay, so tracking is robust. If you look, are there endogenous systems that do this? It seems so, and we found one such system, and that is a system that employs, actually, it's a sigma 70, which is an E. coli housekeeping sigma. There's another sigma factor, it's called an anti-sigma 70, that people had discovered binds very tightly to sigma 70 and sequesters it away from binding to the genes. And so, as it turns out, the anti-sigma is expressed, is expressed by RNA polymerase bound to sigma 70. So it seems that this is one system in nature that seems to employ this antithetic feedback control property. And we only found this afterwards, after one of the reviewers for the paper said, well, okay, this is great, but does this really work in nature? And I actually had no idea. So I called up Adam Arkin from Berkeley, and I said, Adam, we want a system that has exactly these properties. And he said, oh, I have exactly the system for you. And he had this, the system you showed, actually, is the A and B, the ultrasensitive system. They're using it not in feedback, but they're using it to build ultrasensitive circuits that act like switches. And so we thought this is exactly what we want. So they have already the sigma anti-sigma system working. And so we got it from them, and then we built it. I'll show you later. Okay. Now I want to come to the part where actually, in some ways, it's quite surprising, right? So I was very intrigued by this when, and initially, it was more like a discovery. We weren't expecting this. So the idea is, that I showed you before, is, okay, deterministic systems or stochastic systems limit to deterministic systems as the volume increases. So let's take our gene expression network with this controller, and let's apply this large volume limit to it. Basically, that's equivalent to saying you take all of these cells with all their membranes and you dissolve the membranes and you just have one big fat cell with the same exact contents. Okay, that's the equivalent. That's when you apply this law of large numbers, that's essentially what it means. You're not changing any of the reaction rates, you're not changing any of the counts, you're just removing the membrane and you have one big cell. Now you have, this cell is actually closer to the deterministic world, so reaction rate equations, deterministic reaction rate equations would apply to something like this. Okay, so here's a comparison. We've already seen that the deterministic looks like this, right? So you have an overshoot, and then you settle down to the set point. Now, what happens if you take the same exact reactions with the same exact reaction rates and just write the ODE? So in other words, ignore the noise. Just look at the deterministic world. Well, if you do that, this is what you get. You get a system that oscillates. So it loses its steady state value. And it's kind of interesting to try to understand what's going on. Here are the dynamics. So this is the deterministic system, the ODE that you would get. And you can see this is the bimolecular part, so where Z1 and Z2 bind together. So you have a nonlinearity here. So this is a stable limit cycle. And the stochastic dynamics for the mean look exactly the same, except because this is a bimolecular reaction, we don't have moment closure. We have this other term, which is the covariance of Z1, Z2. That's the only difference. Okay? And whereas this covariance is something we cannot compute analytically, we know from the ergodicity theorem that I showed you that this system will always be stabilizing. So these second order dynamics are actually adding damping to the controller. And if you just look at the pure deterministic system, it will be oscillating like this. Okay? So it's a very interesting role for noise. So if you're in the business of building control system, this is great. This is a guarantee that we have that the noise is actually helping you to stabilize. Okay? Yeah?
I'm wondering, so in the first case, all the chemicals that couple are are common to everyone. Where in the second case, what you show is the population average.
Yes.
So isn't it that it's just dephasing between the compounds?
Yes, yes. I mean, in some sense, there's a lot of that going on. This is the next part of my talk. Yeah, exactly, called phase diffusion or dephasing. Yeah, and we'll apply it to oscillators also in a second. But anyway, so this is good if you're building control system. And actually, I gave this part of the talk exactly, I think, in this room about a year ago, right? So the work wasn't published yet; it was still fresh. And there was a smart lady sitting somewhere there, and she said, well, wait a minute, but if this is an oscillator, then this is not a good thing. If you're looking, have you looked at this in the sense of biological oscillators, what this noise does? Because that basically destroys the oscillation. And so, of course, as it happens, we ran into an example that actually forced us to answer this question. But what I didn't know is that this lady was Elizabeth Blackburn, who was the Nobel Prize winner, was sitting there. Remember this? OK, so nobody had told me this. But in any case, it was exactly the right question at exactly the right time. And it actually led us to, in part, let us look at the next part of the talk, the last part of the talk, is how does noise affect oscillations? Right? Before I go there, I want to say that we actually built this system, again, with the sigma anti-sigma molecules from Adam Arkin's lab, and it seems to be working quite nicely in bacteria. Okay, this is still unpublished work. Okay, so the same story now. We're going to look at a different twist. So indeed, this noise is not good news if you're in the business of making oscillators, right? It's good if you're a control engineer, but if you want to make oscillators. So let's look at this scenario and look at the very simple, well, it doesn't have to be simple, just a limit cycle oscillator and see where the problem lies. So suppose we have nonlinear dynamics that has a periodic orbit that is stable, so a limit cycle, basically. And if you look at the dynamics of the phase, what you would notice is that, if you call this the phase, is that they're marginally stable, right? So there's no restoring force to stabilize the phase. And so indeed, if you add some noise to an oscillator like this, noise usually changes the phase randomly, and I think it just kicks the system one way around, the next system in a different way, and as time goes on, and I think this was also alluded to in the previous talk, the noise is distributed everywhere, and then when you average, you get zero, right? So you wipe out, at least you get some constant, you wipe out the oscillations. Okay? So indeed, if you're building oscillators and you have noise, this is not good news, right? So how do you get around this? Well, one way to get around this is to couple the oscillators. And that's, I think, something that people have noticed and studied for a long, long time. The SCN above the eye also does this. And so if you have the coupling, then you can get around a lot of these issues. But here's something interesting. Let's say we start with this periodic system, and then we apply some new input to try to entrain it, right? So you can try to apply an external input with a slightly different frequency. Let's say omega f. That's the forcing frequency. Here's an axis that shows the frequency versus the amplitude. So if omega f is different from the natural frequency, what happens to the output in the steady state? Everything I talk about is in the steady state. Does it entrain, or does it follow this frequency, or does it do something else? Well, it turns out this is a problem that people have looked at for a long time, since the days of Arnold, and it depends on, the answer is it depends. It depends on where omega f is, it depends on the amplitude, depends on the frequency, but roughly speaking, it's something like this. So if the amplitude is sufficiently large, then the output will be entrained. If the amplitude is same frequency but is smaller, it doesn't. Okay? This is deterministic now. Everything is deterministic I'm talking about. And as it turns out, there's some region in the frequency amplitude space that defines the parameters that lead to entrainment. And that's called an Arnold tongue. Okay? Of course, this is one-to-one entrainment. One can also look at 2 to 1 or 1 to 2 or whatever. There are other regions that are more complicated in this plane, but let's just focus on this one simple behavior. Okay, very good. So that's the Arnold tongue. What happens if you add noise? Well, if you add noise, it turns out that, at least from everything we could see, not much happens, right? So if you look at cells that are entrained and you add some noise, they more or less remain entrained. And of course, one has to define what entrained means when you have a stochastic regime, which basically means the vast majority of the power lies at the forcing frequency and its harmonics, right? So that's what we define it as. If it's not entrained to begin with, it's not going to become entrained with this. At least this is our observation, right? For the vast majority of things, there were very small special cases for which this was not true, but I will show you in a little bit. They're almost negligible. Okay, very good. So what's the story then? Noise does nothing. But there is something interesting that we kind of stumbled into. Let's discuss it using a very well-known oscillator, the van der Pol oscillator, right? So if you take a van der Pol oscillator, and now you introduce some noise to it, and then you have a forcing function that's sinusoidal. Again, if this is outside of the Arnold tongue, it will not be entrained. It would look something like this. So this is not periodic. And if you add noise, and you look at the realizations of different cells, you can see that neither one of them will be entrained, but you can also see this moving around of the signal. The interesting thing, which I think is new, and that I would like to report here, is that when you average such unentrained signals, you get something very nicely entrained. Okay? So this is surprising. It's not like these are sinusoidal signals with white noise added to them. So when you're average, you're removing the white noise. If you look at their power spectral density, it's all over the place, right? It's not just here. But somehow, when you add all of these signals together, you get something that is a pure tone with a frequency of the input. So this is what we call noise-induced entrainment. And these are, importantly, uncoupled oscillators. So let's see what happens if you look at the Arnold tongue for the system, right? Here is the Arnold tongue. The green area is entrained, and so deterministically entrained. When you add noise, well, there will be some cells where the entrainment loses along the boundary, where you lose entrainment because of the noise. But it certainly doesn't seem to be helping entrainment of single cells. So noise doesn't seem to be helping entrainment of single cells here. On the other hand, if you look at the population, you have this entire new region here that's in blue that's only entrained with the noise, right? At the population level. So when you average these signals, that's when you get entrained. So there's an expansion of the entrainment region that is due to the noise, provided you look at the population average rather than the single cells. With single cells, not much happens. So that's what we call noise entrainment. So this robustifies, noise appears to robustify entrainment of populations of these uncoupled oscillators. Here's the same phenomena, very different system. This is NF kappa B. This is a model of NF kappa B that's called the spiky NF kappa B model by, I guess, Jensen and co-authors, 2006. And here, basically the same story as before. There's a very narrow region where noise, where noise appears to help, but actually it's also dominated by other regions where actually noise loses you lose the entrainment, right? On the other hand, if you look at this average for the population, again, there's a large expansion of the space. Okay, so let's look at a system with the parameters that are in this new region. So deterministically unentrained, but stochastically is. If you look at, this is not a periodic signal. If you do its power spectrum, if you look at signals with noise, they look like this. And if you average a lot of these things, again, as before, you get a nice average equal to the input. This is the circadian oscillator. This 16 species, 52 reactions. And here we didn't even do the stochastic simulation, the SDE. We just did Monte Carlo simulations, the SSA algorithm. And the same story here. We have new regions of the parameter space where noise actually is responsible for entrainment. Noise for the single cells seems to destroy entrainment for even more cells for this system, right? Okay, and same story as before. You know, single cells without noise, deterministically unentrained. With noise, deterministically, they're still unentrained, but you average, you get the same behavior. Okay, so what's going on, right? Why is this happening? So I want to end on this part because this is, I think, leads to the crux of the matter. Why noise appears to be actually interacting with the dynamics in a good way, you know, to have some additional benefits. And to do this, actually, it would be next to impossible, at least for us, to analytically evaluate something like the circadian oscillator, or even the simple, the other oscillator. So we look at a very simple oscillator, a two-dimensional oscillator called the lambda omega system, that actually, when you transform it into polar coordinates, it has a particularly nice and simple form. So for example, here, this is the r, the radius, theta is the angle, and so dr dt is simply given by this function r times lambda of r. And then this here doesn't depend on theta, it only depends on r. So if you think about the fixed point, so if you find a solution that makes omega at that solution 0, that will be the radius of the oscillation, and then you plug it back in here, that gives you the phase. Right? So that's a very nice, clean oscillator. And if the derivative around this r is negative, you know it's a limit cycle, so it's easy to analyze. OK, so we want to take a system like that and now introduce noise to it. So this is just simply a zero mean unit intensity white noise. And it's introduced particularly to omega here. And now when you look in polar form, it looks like this. Significantly, this noise doesn't affect the R dynamics, so those are still deterministic, in fact. But if you look at the phase, now the phase is impacted by this white Gaussian noise, right? And now if you write down the dynamics of the phase for large T, we can write this down, is simply, this is a constant term. There's a term that grows linearly with time, and then Brownian motion, right? So the phase diffuses like Brownian motion, and this is a phenomena that people have also studied. Is called phase diffusion, right? So this diffuses like Brownian. So if you look at, if you simulate some of these phases for a few cells, you can see that initially you may start with 0 phase, but then as time goes on, you know, this diffuses like Brownian motion, and for large times, the phase is actually uniformly distributed between 0 and 2 pi, which would explain why when you average a bunch of such oscillators, so if you average a bunch of oscillators whose frequency is the same and phase is between 0 and 2 pi, you're going to get 0. So you wipe out the oscillations. And that's what's happening due to the noise, at least to the phase in this case. OK, so that explains perhaps also why our control system works nicely, because the different phases seem to be canceling each other out. OK, here's a noise-free deterministic simulation. And then when you add this noise and just average a few cells, you can see that exactly what you would expect, right? So noise diffusion kills the oscillations. Oops, that's not what's supposed to happen, but OK. Anyway, let me just, might as well put this up. Oh, actually, this is the slide I want. But that's without forcing. What happens to the forcing, right? So what does the forcing do? Now we introduce a small forcing, so weak forcing with a new frequency, omega f here for sine and cosine. And then the system is already nonlinear, it's difficult to analyze, so we want to look at perturbation analysis, right? So this allows you to look at least what happens to the first order terms in terms of epsilon, right? So for small forcing functions, right? And then when you analyze this system, you take this, you take the derivative, substitute back, and solve for the various parameters, you find that this term here that's of order epsilon consists of two parts. One part is has a natural frequency, oscillates with a natural frequency, and the other part oscillates with the forcing frequency, okay? And significantly, so we already know that this part with the natural frequency, this phase is uniformly distributed. We've already seen this. But significantly, the phase that's associated with a forcing frequency is very different, right? So this behaves like a mean reverting Ornstein-Uhlenbeck process, and essentially the phase is focused around the mean, okay? So that's very different from what happens to the phase corresponding to the natural frequency. And I think, as you will see, this is going to be key to explaining this behavior that we see. So now you take, this is just x1. If you add it back to x0, which has the frequency omega naught, then you get this term. So simply, this is epsilon times r plus 1. So you get this term. The phase here is uniformly distributed. The noise, the phase here is actually focused around some specific phase. And OK, that's the same thing. And now, if you look at what happens to the single cells, it's very easy to see. So here's a single cell behavior. So that's just a single trajectory, right, with a particular omega. Now, because this here, you have a 1 here and epsilon there, this is dominated by the natural frequency, right? So the natural frequency dominates the single cell behavior. This is the forcing frequency term. It's multiplying epsilon. It's fairly small, right? Now, but what happens when you average such a thing? Well, when you average such a thing, you basically are averaging each of those terms. And basically, this term, because of the phase distribution, goes to 0. Okay? And remember, to amplitude-wise, this is the bigger term. But its phase is uniformly distributed. This term, which is much smaller, but its phase is focused around a particular point, does not go to 0. And it actually goes to something like this. And it turns out to be the dominant term. And what is its frequency? Well, that's omega f. That's the forcing frequency. So we think at least that's one way to think about this noise-induced entrainment that we see in this system. Now, just to verify, we applied this into these parameters to the lambda omega system, and we look at the deterministic trajectory. Sure enough, the natural frequency is dominating. At single cells, same story. But when you average these things, you get that the forcing frequency is actually the dominant frequency now. So supporting the you've seen before. And now we go back to all these 3 examples and look at these systems that are entrained by the noise. You see what I described earlier. The natural phase is actually uniformly distributed, but the phase that's corresponding to the forcing frequency in all these examples is focused. Right? OK, this is really, I think, the last slide. So basically, I kind of told you the story backwards. OK? So we saw some data that were generated by a colleague of mine at ETH, who was actually a microfluidics guy, and he was applying these sinusoidal inputs to this system. So he was applying oscillating TNF alpha to and looking at the nuclear translocation of NF kappa B. So with this microfluidics device, you can oscillate the TNF alpha concentration, and then you look under the microscope at the nuclear translocation of or nuclear NF kappa B, and he knows that when you have a constant input, there's a natural oscillation, natural frequency, but then as he moved the frequency around in both directions, it will be entrained for a while, and then after some time, it loses entrainment on both directions. So we took some of these cells on both directions that lose entrainment from the right, from the left, and you see about 50 of them here. The blue just gives one example of one of those signals, and same thing here. And then you can see that there's absolutely nothing periodic about those. Those are not entrained, right? So they would not count as entrained. You look at the power spectral density of those signals, and it's everywhere. But then you average these 50 cells, and you get something much more periodic, right? And in both cases. In fact, this is what kind of led us to study this phenomenon in the first place. It's exactly this plot that I'm showing you here. It didn't look like it should be happening, that you average a bunch of these things, that you actually get something periodic with the frequency of the inputs. OK. This is irresponsible speculation at this moment for this slide. So how could this be useful, or is it in biology? Is it used in biology? So we haven't tested any of this, but it's completely speculative. It's sort of, plausible. So as I told you, it's known that the circadian oscillator is coupled, right? And of course, this is entrained by the day and night cycle, right? So you get a very nice entrained signal here. And these rhythmic signals are actually transmitted to the rest of the body through various cytokines, temperature, various other metabolites. The other cells in the body, so these are the peripheral oscillators. Some of them is actually not known that they're coupled, right? Yet their cumulative effect in the tissue appears to be entrained, right? And so imagine now every one of those cells secreting something like, let's say, adrenaline. And if one of these clocks are actually not entrained, but they're all seeing the same sinusoidal cue from the rhythmic signals, and when they get pulled in the blood, that's exactly averaging. And so that could explain why, when you look at the output, it's actually nicely entrained to the rhythmic signals. But again, this needs to be verified at this point. Okay, so conclusions. We've talked about feedback control, and in the presence of noise, one has to be more careful. But also, it turns out that there's a handsome payoff when you use low copy number things for control, because you can get damping and good regulation properties that simply don't exist without the noise. And similarly for the oscillations, right? So I guess the common lessons from this is that these interactions between noise and dynamics often lead to unexpected benefits, but that this behavior, it's difficult to explain, or maybe it's difficult to even see, and is counterintuitive when you look at a single cell level. But when you look at the population level, right, then a lot of these things start making sense again. At least for those two examples that I showed you, that is the case. Okay? And there's other evidence by other people who have showed something similar. Okay, I think this is a good point to stop, and I don't know how much time we have, but I thank you for your attention.